Our next speakers are two. We'll start with Dr. Karakash. She is a maternal-fetal medicine physician and associate professor of obstetrics and gynecology here at Stanford, who oversees perinatal services at Natividad Hospital. She is going to be speaking with Dr. Sonnenburg, who is a research scientist also here at Stanford in the Department of Microbiology and Immunology with a very impressive track record of publications. Together, they will be discussing their collaborative work on maternal and offspring microbiome. So welcome to you both.
Great, I'll go ahead and start. Let me share my screen. Can everyone see okay? Yes. So this is a study where we're interested in a nutritional intervention and how it can affect the microbiome during pregnancy and potentially the microbiome of the newborn infant as well. This study is really only possible because this is a collaborative group where we have Christopher Gardner's group helping with the dietary intervention. On the Sonnenburg lab side, we're doing a lot of microbiome and other types of immune status measurements. And then with Scarlett, we have the clinical side to help us work with the moms too. So I want to start with just a very quick introduction into the microbiome. This is an image that we took in our lab. It's a cross-section of a colon from a mouse that's been colonized with a human microbiota. And what you can see in the top left is, all these little jelly beans. These are all microbes. And so I hope that you get a good sense of how dense this community is. And in the bottom right, we've stained the nuclei of the intestinal cells. So that's sort of the host side. And between these two is the mucus lining, and that serves as an important barrier between our microbial cells and our human cells. We have trillions of these microbes. We have 100 times more genes encoded within our microbiome than within our own human genome. And the field is rapidly progressing away from questions of just who is there towards more of what are these microbes doing. The microbiome is very interesting to lots of people because we've realized now just how connected it is to all facets of our biology. It's been connected to things like obesity and metabolic disease, immune system, and central nervous system as well. And so because the microbiome, unlike our human genome, is quite malleable and adapted to our diet and lifestyle, we view this as a very powerful lever on host biology if we can just understand how to manipulate this community towards better health. So just to give you a brief intro into the gut microbiome, we mentioned that, it's colonized within our gut, but really most of these microbes reside within our large intestine or colon. And the microbes that reside in the colon really rely on dietary fiber for their sustenance. So the human genome doesn't encode almost any capacity for degrading complex carbohydrates found in things like fruits and vegetables, legumes, those kinds of things. So those complex carbohydrates reach the colon largely intact, and that's where the microbes can ferment these complex carbohydrates and generate a multitude of small molecules, including short-chain fatty acids. Many of these small molecules we know can enter circulation, and that's why the microbiome can have such a profound impact on sites distal or away from the colon. We know that the industrialized diet doesn't have a lot of dietary fiber. We eat a lot of simple carbohydrates. Those are easily absorbed earlier in digestion, which leaves the microbiome with very little sustenance. And in the absence of dietary fiber, the microbiome is really forced to rely on the only carbon source that remains, which is the complex carbohydrates within that mucus lining that I showed you earlier. And so in mouse studies, we've demonstrated that when you remove dietary fiber, the mucus lining gets degraded by the microbiome, and that can lead to increased inflammation. And we think that this increase in inflammation could be at the root of many of the diseases that are common in people that eat industrialized diets. We also know that our environment is becoming more sanitized. Water is a lot more sanitized than it has been just from an evolutionary standpoint. And while we don't want to expose ourselves to potentially pathogenic organisms, we think that fermented foods and the live microbes contained within them could be a safe way to reintroduce environmental microbes into our system. And this would be, a way of compensating for the lack of microbial exposure that we have, and would be something that from an evolutionary standpoint, our biology is expecting. So our question within the lab that we're very interested in is understanding whether we can repair the microbiome and improve health. We know that people that live industrialized lifestyles, eating processed food, hypersanitation, overuse of antibiotics, that these individuals have low microbiome diversity, so very few species of microbes relative to non-industrialized individuals. In addition to a low diversity of microbes, we also have low microbe density, so just less overall numbers of bacteria residing within our gut, and also increase in inflammation. And so we're interested in whether the reintroduction of microbes through fermented food, or maybe more directly, or changes in diet can improve our microbiome and therefore improve our health. So we performed a study, a clinical trial a few years ago called FIFO for fermented and fiber food. And what we did was take 36 participants. We randomized, these were just healthy adults. We randomized them to either a high fiber diet or a high fermented food diet. We took a bunch of stool and blood from these individuals, and we did very intense microbiome profiling and also immune profiling too. And this was published now a couple of years ago in Cell. We found that participants were able to successfully increase their fiber and fermented food intake. So at the beginning of the study, participants were eating a little, roughly about 20 grams of dietary fiber per day, and they got to over 40 grams during the maintenance or highest point phase in the study. For the fermented food serving, they started at 1 serving or less per day and got up to about 6 servings of fermented food per day. So people did, this intervention very well. And so if we just start by looking at the fermented food arm, what we found was that participants that were consuming the fermented food increased their microbiota diversity. They started out at the beginning of the study with roughly 100 different kinds of species of microbes that we could detect in their gut. And by the end of the study, they had closer to 130 different species of microbes within their gut. So full, almost third on top of microbiota diversity that they gained during the study. We tested to see if this increase in diversity came from the fermented foods. Was it just the bacteria that they were consuming? And the answer to that is no. So we don't, at this point, really understand where that diversity came from, but there was just something about consuming fermented food that made these participants' microbiomes receptive to retaining new species. While the diversity went up, we also saw serum cytokines and chemokines decline across the whole cohort. So this was just a very profound lowering of the inflammatory status of the participants consuming the fermented food. For the high fiber food, I don't have all the data here just for the lack of time, so I'll just summarize what we saw. Interestingly, we saw that people really improved their overall diet despite the instruction of just eating as much fiber as possible. That's what we told participants. And by doing that, they shifted their consumption of protein from mostly animal protein to mostly plant protein, which is beneficial. They also improved the overall nutrient profile. So they just ate a lot more vitamins and minerals. They ate a lot less things like sodium. So just in general, their diet improved. We didn't see an increase in microbiota diversity like we had seen in the fermented food arm, but we did see an increase in microbe density. So there were not more species, but just more overall bacteria total within their microbiome. And we did see that their microbiome increased their capacity for degrading dietary fiber. So that was an indication that the microbiome was responding to the increase in fiber consumption. And the immune response, we saw it more of an individualized response, and we're currently following up to try to understand what factors impacted the immune response to fiber. And so now we're interested in understanding whether the consumption of high fiber foods and or fermented foods could improve the microbiota and the health of pregnant women and their babies. So now I'm going to transfer this over to Scarlett, who's going to give you more details on the study that we're doing for that.
I have to unmute myself first. Sorry about that. Okay. All right. Hi, everyone. I'm going to start with Erica's last slide. After everything we know about how the diet can influence potentially the microbiome and the immune markers, the question for us is, can the consumption of high fiber foods and fermented foods improve the microbiota of the mom and the baby? And I'll talk briefly about why we even think that a pregnant person's microbiome could affect their fetus and their newborn. So the idea of the developmental origins of health and disease, otherwise known as the Barker hypothesis or fetal origins hypothesis, is that the intrauterine environment, maternal conditions, and early exposures influence the fetal and the offspring's health. We know that pregnancy complications like fetal growth restriction or overgrowth, maternal diabetes, hypertension, and obesity lead to long-term consequences for the offspring, such as cardiovascular disease, hypertension, diabetes, and obesity in adulthood. So studies suggest that the early microbiome, meaning in utero and in the first years of life, have an important role in the development of the offspring's short and long-term health and immune development. Some potential mechanisms are microbial metabolite exposure in utero, like the short-chain fatty acids that Erica spoke of, and different exposures at birth triggering a fetal or neonatal immune response that helps immune development. I'll briefly talk about what we know about the microbiome in normal pregnancy. So in terms of the intestinal or the gut microbiome, the data we have is limited and with mixed results. Several studies report gestational age variation, meaning in the first trimester, it's similar to a healthy non-pregnant controls. And by the third trimester, the microbiome is more similar to disease-associated dysbiosis. And in terms of the evidence for adverse pregnancy outcomes, like gestational diabetes and preeclampsia, the gut microbiome changes are also limited and with mixed results. This group probably knows a little bit more about the vaginal microbiome, and normal microbiome in pregnancy is lactobacillus dominant. And in the case of the vaginal microbiome, as opposed to the intestinal microbiome, lower diversity seems to be better. So lactobacillus poor environments or more bacterial diversity, like you have in BV or bacterial vaginosis, has been associated with preterm birth. Recent studies are looking at how the maternal microbiome affects the offspring's health and include an increased risk of preterm birth, like I talked about, and its long-term consequences. The intestinal microbiome changes have been associated with neurodevelopmental outcomes, metabolic disease, and altered offspring microbiome. So many factors shape the neonatal microbiome, like mode of delivery, antibiotic use, diet, breastfeeding, environmental exposures, and the maternal microbiome, which is what we're studying, and specifically how the diet influences it. So our study aims to investigate whether modifying the pregnant person's diet can affect their microbiome and how that maternal microbiome influences the infant's early microbiome and immune development. Ours is the first, as far as we know, randomized controlled trial of dietary intervention in pregnancy looking at this. And our hypothesis is that the pregnant participants who increase their fiber and or fermented food intake during pregnancy will not only increase their own microbiota diversity, but also their infants. Where 133 participants were randomized into 4 groups: high fiber, high fermented foods, high fiber, and high fermented foods, and no intervention, which was the control group with who maintained their current diet. The inclusion criteria are those. And the exclusion criteria are many, and that's because we're really looking for general healthy pregnant participants, but some common exclusion criteria are these: BMI greater than 40, pregestational diabetes or history of gestational diabetes, gastrointestinal disease, severe hypertension, certain medications, history of preterm birth. This slide shows the study timeline during pregnancy, and home samples include microblood samples, vaginal swabs, and stool samples. And there are 4 time points: 3 during pregnancy and 1 postpartum, when the participants need an official blood draw done. And throughout the study, the participants will be regularly meeting with for dietary education with an amazing team of educators, as well as taking some surveys along the way. The postpartum timeline includes breast milk collection and infant stool samples. The patients that do deliver at Stanford also have cord blood samples collected. There is an infant heel stick at 5 months. Our recruitment is done, so that's the summary of what we're doing. We recently finished recruiting. Sorry for fast talking, but I know we're behind in time. Thanks for your attention.
Thank you both so much for presenting all of that. Amazing work. I would encourage anybody to put questions in the chat, and just in the interest of time, we'll move on to our next speaker, but we can either answer them at the end or offline. So please put them there. Thank you both so much.